Hello everyone, today we'll talk about peptidoglycan. So peptidoglycan, it is present in the bacterial cell wall and it is also known as murine or mucopeptide. It is present in gram-positive as well as gram-negative bacteria. In gram-positive bacteria cell wall, it is a major component, whereas in the cell wall of gram-negative bacteria, it is a minor component. It forms a strong covering around the bacterial cell wall and thus prevents swelling and the cell lysis of the bacterial cell. Lysozyme which is present in egg white, tears, saliva, sweat. So it cleaves the glycosidic bond present in the peptidoglycan and thus lyses the cell wall. It was first discovered by the Fleming in 1932. Penicillin, it is a very commonly used antibiotic. It inhibits the cell wall synthesis by preventing cross-linking in the peptidoglycan. Now coming to the structure of peptidoglycan. Peptidoglycan, as the name suggests, it has two parts, peptide and the glycan. So glycan part is made up of two monosaccharide, NAM and NAG. So this is the repeating unit present in the peptidoglycan. There is linear chain of these polysaccharide which are cross-linked through beta 1 4, which are the glycosidic bond present over here is beta 1 4. These are linear chains and there is no cross-linkage between these polymer chains. They are cross-linked via the peptide chain. So this is a heteropolysaccharide. Heteropolysaccharide because there are more than one monosaccharide repeating unit. And the repeating unit is NAG and NAM joined through a beta 1,4 glycosidic bond. Now NAG stands for N-acetyl glucosamine. And NAM stands for N-acetyl muramic acid. We had talked about these structure in our previous lecture, so you can go back and look there. So this is an amino, both of them are actually amino sugar, and it is a linear polymer. It is a linear polymer, or we can say unbranched. Now this cross linkage is by it. These linear chains are cross-linked by peptide. To this NAM, to the NAM, they are attached 4 amino acid. So it tetrapeptide is attached, and the sequence of this tetrapeptide is L-alanine, D-glutamic acid, L-lysine, and D-alanine. So in case of this amino acid, in case of gram-positive bacteria, it is L-lysine, or in case of gram-negative bacteria, it is mesodiaminopamalic. Mesodiaminopamalic acid in case of negative bacteria. Now the cross linkage. How this cross link? The cross linkage. There are two types of cross linkage. Direct. The cross linkage is is either direct or indirect. Indirect cross linkage, this the cross linkage, the two peptides are linked by a pentaglycine chain. So here uh, this third amino acid, L-lysine of 1, is cross-linked with the fourth amino acid. So this L-lysine is cross-linked with the D-alanine via a pentaglycine in an indirect method. In direct, this is this is directly linked with this via peptide linkage, and in case of indirect, it is cross-linked by a pentaglycine bridge. Now, we have talked about in the previous slide about lysozyme. Where does this lysozyme act? This lysozyme cleave this beta 1,4 glycosidic bond, and the penicillin inhibits cell wall synthesis. It is an antibiotic. So penicillin inhibit cell wall synthesis by inhibiting this cross linkage. So thus it makes the cell wall weaker. This is all about peptidoglycan, and please do share this with your friends, and please do subscribe my channel. In the next lecture we'll talk about glycosaminoglycans. Thank you.